So in the previous video, sorry, I'm trying to fix this. In the previous video, we spoke about normal DNA sequences. So when there are some type of mutation or change to the DNA sequence, it is going to change the proteins that are made. So a point mutation is a change of one nucleotide base. All right, so in this case, this one used to be a C and it got changed to a T. All right, so when you read this, it's still 3 codons. So this is going to be U, A, C, A, G, A, G. All right, the transfer RNA is going to be A, U, G, U, C, U, C. All right, so when you read this, you still read it in the 3 codons. These three, and these 3. All right, so if we look this up for the amino acid, we have U. We have Phi still, that did not change. Then we have UAC, a U, an A, a C, TYR, AAA. We have an no, got wet. We have an A with an A and an A. That will be a lysine or lyse, and then we have a GAA, a G, the A, and that is a glue. So if you look at the original paper, it was a glue and a glue. Because of the point mutation from that C to a T, it changed it so that it was a lyse now. All right, so that's a substitution. For frame shifts, we have insertion and deletion. Basically what it does, it actually moves the frame. When you have an insertion, it moves the frame out, adding to the amino acids that are there, or the amino acid the DNA sequence, sorry. And then for the deletion, you're picking one up, so basically it's squeezing it together at the end. All right, so when you have the messenger RNA for this one, it is going to be I'm left-handed, so it's a little U, A, C. This is going to be a G, A, G, and A. All right, you're still going to read it with the 3 codons. So the anticodon is going to be A, U, G, C, U, C, U. All right, so when you read this one, you're going to have, I just want to get the paper. The amino acid sequence is going to be A, and A. It's still going to be a lyse. Oh, sorry, wrong one. U. That will be a Phi. UAC. U with the A with a C. That has not changed. Thymine. Now we have a GGA. G with a G with an A. It's a gly. And then we have an AGA. A with a G with an A. All right, I'm going to go over deletion and then I'll explain the actual chart from the PowerPoint. All right, so for the messenger RNA, I shouldn't have moved that. Sorry about that. We have U, U, U. So there used to be an A here, but the A got taken out, and what happens is it actually squeezes the whole DNA sequence in, because that amino acid or that nucleotide is missing. So since it was taken out, all the DNA sequences moved inwards. So this C would have been the next nucleotide in the sequence, so it gets pulled in. So that is from outside. All right, so we have the T with an A G with a C, A, this is going to be a G, this is going to be an A, G, and a G. The transfer RNA will be A, U, G, C, U, C, U, C, C. All right, same thing. We're going to underline the messenger RNA. 3, 3, 3. There's still 3 codons. All right, so when we read this, we still have U, which would be a phi. Then we have an ACG, an A with a C with a G. Then we have AAG, A with an A with a G as a lyse. And then we have the last one is AGG, A with a G with a G, and it's an ARG. All right, so the whole purpose of this is, let's say we're making a protein for hair, or any type of protein. It could be a specific amino acid as well. Not amino acid, antibody, sorry, my bad. All of those are made out of proteins, muscles. So if the DNA sequence is wrong for any of those proteins to be made, they will not be made properly. The same thing can be said for bacteria as well. So when you look at this sheet, this was the normal sequence that was needed, but because of the substitution in one amino acid, it changed the actual sequence. So let's say this was for an antibody, that antibody may not work. I'm talking about humans. All right, so the other thing that you may want to think about is in terms of antibiotic resistance. If there's a mutation in one nucleic acid in the bacterial DNA, it can change it so that it can, this protein now all of a sudden can make it resistant. I'm not saying that it will make it resistant. It's just trying to get you guys to think on a different like level, or think broader, because the antibiotic resistance is a very it's a very complex topic. So I'm just trying to get you guys to think outside the box a little bit. So when you have an insertion, you're inserting the C here, but you're moving the whole sequence over. So everything, if you noticed, all of these started with a phi. These were a tree or tri, whatever. And then it's only where there was a change in the nucleic acid is where it starts actually changing. For substitution, it's very simple. It's only changing one. So it changes the one amino acid. The problem with this is, let's say it was supposed to be a stop codon here, but it stopped here. Because of the substitution, it makes the amino acid shorter. That can be a problem. So then when you're inserting a new amino acid, you're moving the whole sequence over to the right. So whenever you read this, it's always going to be 5' to 3' direction. All right, always, no matter what. All right, so when you have insertion, you're inserting a new nucleic acid, nucleotide, same thing, and you're moving, or the shift, the frame shift is moving the whole thing to the right. When you have a deletion, for this one, we're taking out the A. So when you take it out, you're squeezing everything over. I feel like there was a price, the Price is Right like thing like that. Like you have to take out a number and it squeezes it together to make the price of the item or something like that. I remember watching it as a kid. It reminds me of that. So I'm aging myself. So when you take out a nucleotide, you're squeezing the rest in. So you're bringing in the whole sequence. So this C that came in, this would have been the next one here, 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 here. Because you took that one out, you're basically bringing in the C that was out here into the frame. All right. Hopefully you guys understand this. This sheet, this one, I prefer this one over this one. This will be on your exam. It will be worth 0 points. Any questions about this? Email me and let me know. Other than that, good luck with the studying. Bye.